So this is the stroma. And here's your thylakoid. We'll stop. Can we be quiet, please? Thank you. We need a protein in the membrane of the thylakoid. This protein is called the PS2. Sometimes we call this also P700 because it does a great job of absorbing sunlight that is 700 nanometers. Inside of this PS2, if you recall, we have green little dots called chlorophyll A, chlorophyll B, and so forth. And the last two chlorophyll A's, these are the most important ones here at the bottom. A pair of chlorophyll A's that do a good job of absorbing 700 nanometer light. Okay? So we also need, to get everything going, we also need sun, right? So our sun provides the energy that is being now collected by those by those chlorophylls, and that energy kind of gets passed back and forth until that energy gets onto those two chlorophyll A's. The other thing that we need though is obviously a source of electrons to energize this whole thing, and that source is going to come from water. So if you remember water, it looks like this. The blue dots are the electrons that are holding this water molecule together. Shared electrons means covalent bonds. So as all this is happening, all simultaneously, this protein has a huge affinity for low energy electrons. So two of those electrons will be stolen from water and they end up here, low energy electrons. As a result of this, we now have oxygen with 2 electrons. And then look what happens. Since this electron was stolen, we now get a big concentration of H plus inside of the thylakoid, right? Low concentration of H pluses out there in the stroma. And because this happens more than once, obviously more than one water molecule, this electron kind of gets shifted here, and here comes another oxygen with its 2 electrons together. That gives us O2, and we now, I'll do everything per glucose. Now we will have made 6 O2s. That's one of our products. Right off the bat, that oxygen used to be part of water. Make sense? So now we have these low energy electrons. Well, because of the sun's energy and the fact that photosystem 2 can capture that energy, we can now energize these electrons. So it's always indicated as them going up a photosystem like so. So here are those 2 electrons. They don't really go up the photosystem, but that shows you that they are now energized. And so for the rest of the time, what I'll do is I'll circle them in red. Okay? Kind of crappy red, but you get the idea. Let's just go orange. Make sense? So now these electrons are glowing. They have high amounts of energy. So very quickly after that, those electrons get now passed down to a set of proteins called the electron transport chain. Sometimes they're also called cytochromes. So chromes, cytochrome complex. At any rate, those electrons now go through these proteins and end up losing their energy. Notice they're not glowing anymore. Now we have low energy electrons. Those proteins have taken that energy of those electrons. Where did that energy go? Well, that energy went towards active transport, which is low to high concentration. So H pluses, or protons, of which we have few out in the stroma, now get pumped from the stroma through here into where we have more of them inside the thylakoid. So that energy was used for active transport, which is pumping stuff from low to high concentration. Make sense? Good so far? So what's the purpose of creating this huge high concentration of H pluses here? Well, that is to send them through another protein called, remember, ATP synthase. All right, so this is ATP synthase. So now ATP synthase, again, think of this as water behind Hoover Dam. Right, lots of water behind Hoover Dam. Open up a channel, absolutely no energy is required. That water will flow through a channel, spinning a turbine, producing electricity. And so this is the smallest little generator, right? Generator in the world. So those H pluses will now flow across ATP synthase back into the stroma through from high to low concentration. It just happens. Right? We don't need to put any energy into that.
If they're already outside, why do they need to come inside and then go outside?
To create the higher concentration. Because otherwise the concentrations would be equal on both sides.
Did they start outside?
No, they start inside because remember they come from water.
All those H pluses?
Yeah.
Oh,
Yeah, so you already have more inside. So this gets even more of them to be inside. That's why we're going from high or from low to high. Okay? So as this is happening, what we can do now is we can now take 12.
Do you want my glucose?
Yeah. So we will make now one. Yeah, let's do that. So look, we will now make. We can now turn ADP plus a phosphate and turn this into ATP. But remember, per glucose, we're going to need 12 of them are going to be made. So let's go with 12 here, and we're going to make 12 here. So this has to happen 12 times. Okay? Does that make sense?
Mm-hmm.
So in essence, we should also have done this. So I will need 6 H2Os per glucose for this to happen, and I will make 6 oxygens. Make sense? Good?
Yeah.
Yes.
Okay, we don't want low energy electrons. So we made ATP, but we don't want low energy electrons. We want high energy electrons. And so these electrons now will travel to another protein called PS1 or P700. Just kidding. This was 680. Nobody's paying attention. Nobody corrected me. You guys aren't.
The height you did correct.
Did you? Out loud?
Just now, you didn't hear
Oh.
it. No.
And so those electrons are down here, and if you remember, we have more green little chlorophylls, like so, and we need more sun to energize those electrons. Let it be noted, there's not actually 2 suns, it's just 1 sun, but I just don't know. I guess I could have drawn an arrow, but you guys get the idea. So the energy gets passed on, and that energy makes it on those 2 electrons, and voila, those electrons become re-energized. Here they are, glowing, but this time we're not going to get rid of their energy. We're now going to carry them to the Kelvin cycle, which means we need an electron carrier. And that electron carrier is NADP+, which picks up those electrons and becomes NADP with
NADPH.
high energy electrons, but now it went from +1 to like -1. It doesn't want to be -1. It wants to be neutral, so it picks up a hydrogen, which is a +1 charge, and now we end up with NADPH, which is
Close.
sweet. That would have caught on camera too.
That would have been good.
Frank, you recording me?
Yeah.
Damn it.
Six people.
6 people? You guys are off. Correct. And this has
Isn't it NADP minus?
no.
So that's some teach.
Now it's an ADPH with high energy electrons.
Before that.
Yeah. So for glucose, we're going to need 12 of these by the light reactions. So 12 ATPs, 12 NADPHs are made via the light reactions. So what you see on the board is the light reactions. Good? Makes sense? Okay, so now let's talk about the Calvin cycle. So again, Calvin cycle is a cycle. You've got to start somewhere. So what do we have? We have a 5 carbon, 1, 2, 3, 4, 5. This thing is called, and you've got 3 of them. They're called RUBP, ribulose biphosphate. So that's already there. Where did it come from? Well, it came from the light or the end of the Calvin cycle. So here you get an important enzyme called rubisco. Rubisco. And that rubisco kind of ensures the following. It's going to make sure that we take in 3 CO2s and they come in. So 3 carbons plus 1, 2, 3, 4, 5 times 3, 15 carbons is going to give us now is going to give us 6 3-PGAs, and each one of these has 3 carbons. Make sense? Very low energy. PGAs do not have a lot of energy, so this is where the payoff kind of phase of energy comes in. So these 12 and these 12, trying to think ahead. Okay, so this is where 6 ATPs give up their energy, and we now get 6 ADPs plus phosphate. And this is also where 6, I know I'm writing 6, but I'll explain this in just a second. This is where also 6 NADPHs give up their high energy electrons. And when something gives up the high energy electrons, it is oxidized. Very good. They are oxidized. They were reduced, right? NAD plus was reduced to NADPH. And this now becomes 6 NADP plus. And what we get is, after that, after all this energy was unloaded, we now have 6 G3Ps, which is a very high energy molecule since it absorbed the energy of 6 ATPs and 6 NADP pluses. So a couple of things just to refresh our memory here. Both this and this, now we'll go back to the light reactions. So here's your ATP and here's your NADPH to pick up more energy. Make sense? So at this point down here, 1 G3P leaves, that's the product, that's half a sugar. So we have 5 G3Ps left over. But rubisco can only act upon our RUBP. And so we now have to supply the energy of 3 more ATPs. Wrong color. My hands are going to look like a rainbow after this. So 3 more ATPs give up their energy and become 3 ADPs. And now we regenerate this. Right? So a couple of things, well, one big thing. So we made half a glucose, correct? So you guys remember that 6 and 6, 12 and 12. So all of this, right? Everything that you see here in this bracket, all of that has to now happen times 2. And that's going to give us 2 G3Ps, correct? And when a G3P, so a G3P plus G3P combined, plus 2 more hydrogens, plus 2 H's, that will give us C6H12O6. And I'm not going to draw everything where they are, but remember, this C6H12O6 sugar, glucose, is packed with those high energy electrons. So the very same electrons that water gave up, I switched the color of my electrons somewhere. I went from blue to purple. You guys okay with that? So this glucose now has the energy of the sun because it has those high energy electrons in it and on it. Make sense? Yeah?
Did you get the 2 H's from photosynthesis?
Yeah, remember this is all happening